In part 1 of this lecture, we discussed how is light energy harvested into producing ATPs and NADPH. In this second part, we shall discuss how are the products of light reaction used for synthesis of glucose. You know from the previous lecture how is light energy harvested into producing ATP and NADPH into the stroma of the chloroplast. Here in the stroma, glucose biosynthesis takes place. Glucose is a 6-carbon molecule. The biosynthesis of sugar is called as fixing of carbon dioxide. Because glucose is a 6-carbon molecule, 6 molecules of carbon dioxide are to be fixed to result in 1 molecule of glucose. This is achieved through what is known as Calvin cycle. This is the final equation. Carbon dioxide along with water using ATP and NADPH synthesize glucose. The carbon dioxide comes from air. ATP and NADPH were generated during the light reaction. Hence, dark reaction depends upon the products of light reaction. So, as long as there is light, dark reaction will continue. After light is no more available, the dark reaction will go on for some time and then stop. Hence, we can say that dark reaction is a misnomer and it is better to call it as the biosynthetic phase of photosynthesis. The water is absorbed mostly through the roots. We know that carbon dioxide is present in the air. How does it combine to get into the biosynthetic phase? In some plants, the first molecule to hold carbon dioxide is a 5-carbon keto sugar known as ribulose bisphosphate, more affectionately called as RUBP. This 5-carbon sugar with 1 carbon of carbon dioxide becomes a 6 carbons, which immediately breaks down into 2 3-carbon products. And these 3-carbon products is the first stable product of the reaction. And because a 3-carbon product is the first stable product of this reaction, we call it C3 pathway. In some other plants, the first molecule to hold carbon dioxide is a 3-carbon molecule, phosphoenol pyruvate, abbreviated as PEP. In these plants, 3-carbon PEP with 1 carbon of carbon dioxide becomes a 4-carbon organic acid, oxaloacetic acid. And this 4-carbon oxaloacetic acid is a stable product. So because the first stable product is a 4-carbon OAA, we call it C4 pathway. Now, whatever may be the first primary acceptor, IUBP or PEP, and whatever may be the first stable product after accepting carbon dioxide like PGA or oxaloacetic acid, C3 or C4, the Calvin pathway occurs in all the photosynthetic plants. Each run of Calvin pathway or Calvin cycle fixes 1 molecule of carbon dioxide. So 6 runs of Calvin cycle will fix 6 molecules of carbon dioxide and produce 1 molecule of glucose, because glucose is a 6-carbon molecule. We have seen there are 2 major pathways for carbon dioxide fixation for glucose synthesis, the C3 pathway and the C4 pathway. The Calvin cycle is seen in both. In fact, C3 pathway is the Calvin cycle. So C4 pathway is eventually followed by C3 pathway or Calvin cycle. Calvin cycle can be divided into 3 major parts. First step is carboxylation. Here, carbon dioxide combines with RUBP, or we can say RUBP is carboxylated. This is the most crucial step because 1 carbon is added and is catalyzed by a well-known enzyme RUBP carboxylase, or more affectionately as Rubisco. O is for its oxygenate action that we shall discuss in the third part of the lecture. Second step in Calvin cycle is reduction. Here, carbohydrate is formed because to 1 carbon of CO2, 1 molecule of H is added. This H comes from NADPH, which was formed during light reaction. So 2 molecules of ATP and 2 molecules of NADPH are used because there are 2 molecules of 3-PGA in 1 cycle. And the final and third step is in which RUBP is regenerated to combine with another. This is the pictorial representation of the Calvin cycle, which begins with RUBP, you can see 5 carbons. 1 carbon of carbon dioxide combines with RUBP, and the 6 carbons immediately break down to produce 2 3-PGAs of 3 carbon each. These are then reduced by NADPH. 2 carbons of PGA are reduced to 2 molecules of triose phosphate. In the final step, triose phosphate 2, they combine to regenerate IUBP, and in this process 1 carbon is fixed. The carbon that was gained from carbon dioxide is fixed. Second rerun of the cycle fixes another molecule of carbon, and after 6 such reruns, glucose is synthesized, which is a 6 carbon molecule, and is removed for further processing. A quick revision of Kelvin's cycle shows IUBP with carbon dioxide fixation produces 2 molecules of 3-PGA, which are then reduced to 2 molecules of triphosphate using 2 ATP and 2 NADPH. These triose phosphates combine to reproduce IUBP, wherein 1 ATP is used. This part of Kelvin cycle is the carboxylation part, where RUBP is carboxylated. This portion of Kelvin cycle is the reduction part of Kelvin cycle, where 3-PGAs are reduced to triose phosphates. And this portion is the regeneration part of Kelvin cycle. IUBP is regenerated. And in the whole cycle, we see after 6 cycles, 1 molecule of glucose is produced using 6 molecules of carbon dioxide, and 6 into 3 molecules of ATP in each cycle, and 6 into 2 molecules of NADPH are used. The C4 pathway occurs in special plants. These plants are adapted to function optimally in hot, dry, and excessive sunlight conditions. To adapt to these conditions, the leaf have special anatomy. They tolerate high temperature, they respond to high intensities of light, and do not undergo photorespiration. And because of this adaptation, have a greater biomass productivity compared to C3 plants. So what is the special leaf anatomy? The vascular bundle of the leaf is surrounded by large cells called bundle sheath cells. This anatomy is called as Kranz anatomy because Kranz means wreath. The bundle of sheath, the sheath forms several tightly packed layers around the vascular bundle, and it looks like a sheath in cross section. These bundle sheath cells, they have a large number of chloroplasts. They have thick cell walls, so the water does not leave the bundles. The vascular bundles do not lose water. They are impervious to gaseous exchange, and there are no intracellular spaces. Example of such plants are maize, sorghum. You can see a picture of cross section of such a leaf. The vascular bundles can be seen. The bundle sheath cells are these. They have a large number of chloroplasts, and mesophyll cells fill the rest of the space. The primary acceptor of carbon dioxide here is PEP, and the first stable product is OAA. Ultimately, in the C4 pathway, the primary acceptor of carbon dioxide is a 3-carbon molecule or phosphoenol pyruvate, shortened to PEP, PEP. PEP is present in the mesophyll cells. PEP combines with carbon to yield a 4-carbon organic acid OAA, oxaloacetic acid. Obviously, this is formed in the mesophyll cells, and the enzyme which catalyzes this reaction is PEP carboxylase, or in short PEPcase. This enzyme obviously is present in mesophyll cells. Once OAA is formed, it is converted to other 4 carbon compounds within the mesophyll cell itself. These can be malic acid, aspartic acid. These new 4 carbon compound cell is then carried from the mesophyll cells into the bundle sheath cells. Once inside the bundle sheath cells, the 4 carbon acid is broken down to release carbon dioxide, which is retained by the bundle sheath cell, and the 3 carbon compound, which is transported back to the mesophyll cells, where it is converted back to PEP. This completes the cycle. So carbon dioxide, which was released in the bundle sheath cells, enters the Calvin cycle within the bundle sheath cell, that is the C3 pathway, to make glucose. As we have seen, the bundle sheath cells are rich in chloroplasts, so the supply of NADPH and ATP is already there. Carbon dioxide is transported to it through this C4 pathway. Therefore, the bundle sheath cells are rich in Rubisco. So, having Rubisco, ATP, NADP, and carbon dioxide. This is a depiction of the cross section of a leaf from C4 pathway plant. We can see this is the upper epidermis, the bundle sheath cells, the vascular bundle, which is surrounded by bundle sheath cells, mesophyll cells. An enlarged view of this part is shown on the right. You can see the mesophyll cell in contact with the bundle sheath cell. Carbon dioxide enters the mesophyll cells, which are loosely packed, where PEP is present. These two combine to produce oxaloacetic acid, which is converted to other 4 carbon acids. This 4 carbon acid is transported into bundle sheath cell, which extracts carbon dioxide to enter fixation by Calvin cycle. We can see large number of chloroplasts, and the 3 carbon acid which is left is transported back to mesophyll cell, where it is converted back to PEP. We shall summarize in the end. The dark reaction occurs in the stroma and results in formation of glucose. The glucose molecules are then removed and converted further to sucrose and starch, etc. for storage. Glucose synthesis occurs through Calvin cycle, where carbon dioxide and water, they form glucose using ATP and NADPH. 2 major pathways for carbon dioxide fixation occurs for glucose synthesis. These are C3 pathway and C4 pathway. C4 pathway is eventually followed by C3 pathway. The extra step is for adaptation to hot, bright and dry conditions. The next part of the lecture we shall discuss photorespiration.